Thanks for your great introduction, Nicole. I'm so happy to give you a talk today, and also a little bit nervous because my English is super terrible. So if you not understand me, just pretend that I am explain very advanced science. So since I joined here March this year with Anthony's lab, yeah, I guess our project is going well, but not enough result or data for a presentation. But I'm happy to introduce you. What I did in my PhD progress is lichen microbiome. So after the introduction of lichen and its microbiome, and I'm going to show you four topics what I did. And the end of the presentation, I can show you what I'm doing in UH. Yeah, introduction. Lichen is a really interesting organism, symbol of symbiosis, and we can typically distinguish by their growth type, like this leaf-like one, foliose lichen, this crustose lichen, and structure like a branch. We can call it fruticose lichen. And this symbiosis between fungi and their protobiont is really interesting. That protobiont can generate some carbon source by the photosynthetic progress. The result is usually sugar alcohol in lichen phallus. Then what these protobionts get from this fungi, this hyphae to act like a shelter for this protobiont. So they can be protected by some environmental stresses like UV or dry condition. Lichen is also important in ecology. They covered about 6% of Earth's land surface, and actually the Asian name of lichen, Jiho in Korean, it means that Ji means land and Ui means close. So literally close of land. And they can do as a pioneer in a succession stage. So with the most lichen come first and grass, shrub and tree. And lichen can be used for a human being for a diverse purpose. Some lichens are used in some base note in a perfume. And we know that litmus paper that change their color in different pH, it is made from lichen. And they can also be used as some food or medicine material, especially in East Asia. So I got my PhD in the lichen lab in South Korea, and most of my colleagues advise that they do lichen taxonomy. But I found myself very soon that I'm not good at it because when do you go field, they say like, look at the color of that lichen is bluish gray. Then what about that? It's grayish blue. And about the microscopic condition, my friend says, look at the size of this species ascospore. It is 2 micrometer. But what about that? 2.2. Different species. So later I got more interested in some diversity or some more like ecological things like a community structure. So here's my friend, lichen microbiome. From a traditional definition of lichen, that symbiosis of fungal microbiome and protobiont, but there are lots of research based on culture or amplicon sequencing base, that now that we know that in the lichen phallus, there are lots of diverse species of bacteria and other fungi. These other fungi, lichen microbiome, they are really taxonomically diverse, and so that they can produce a diverse secondary metabolite, and they are regarded as doing some ecological role in lichen phallus, like nutrient cycling or promoting host immunity. And there are also some confusion of using terms of endolichenic fungi and lichenicolous fungi. Lichenicolous fungi make a specific structure on the lichen, so we can see visually with our own eyes, but the amplicon sequencing results show that even in a healthy lichen, so there is no symptom, but it also contains a lot of DNA belonging to lichenicolous fungi, so it might be better than using lichen microbiome, not in endolichenic or lichenicolous fungi. So the first topic is culture diversity of lichen microbiome. So in that time, there were not many reports about the culturable lichen microbiome. So I was wondered that how can I isolate super diverse fungi of this lichen? So I set these 4 variables. So which parts of lichen and how much the degree of surface sterilization, and what about the lichen segment size, and also use some different kind of media. So I collected 5 lichen thallus belonging to one species named Palmotrema tinctorum, and I got about 1,700 isolates and belonging to about 100 species. And the rarefaction curve showed that my sampling effort is quite enough. Right. And the isolation process, it was like a war against the fast-growing filamentous fungi. So it means that I can see some slow-growing fungi on the media. They say it's like I'm a noble species, isolate me. But in the larger cases, they're covered by some other fast-growing hyphae, they say, not in this time. So when I used a small segment with maybe lower CFU value, and using a combination of such a media like nutrient-rich media, like potato dextrose, and nutrient-poor media, like basal bolus medium, so using that approach, I can isolate diverse fungi, including the slow-growing one. Then the next topic is just comparing the diversity with amplicon sequencing. Yeah, I developed the isolation method, so I just wondered how much this cover result can cover the amplicon sequencing result. So in one phallus, 5 segments for the amplicon sequencing, and another 5 segments for the culture. So as expected, there are higher diversity in amplicon sequencing and about taxonomic composition. Even though I was successful to isolate some slow-growing fungi belonging to this Docidiomycetes and Urothiomycetes, but the dominant taxa was Sodariomycetes, but Docidiomycetes in a metabarcoding amplicon sequence. Then what about the overlap of these two approach? I found that 37 OTUs are commonly observed in these two approach. And as you can see, I got some kind of candidate of novel species to identify to or higher level of taxa like order level, class level. But what is the most interesting part is here. I just expected amplicon sequencing data to cover all the results of cultural results, but still don't know why. Maybe because of PCR bias or some primer problem. Don't know why, but it is says like that amplicon sequencing is not always better than culture in an investigate diversity. Maybe they can be some kind of complementary approach. And found that in the amplicon data, I found if we want to investigate some diversity of lichen microbiome, we don't need a really large of sequence numbers because it was really good news because you know that lichen host is fungi and microbiome they are fungi. So I need a step for a filtration of host sequences. So yeah, good news. The next topic is unique structure of lichen microbiome. So yeah, there is a lichen microbiome, but what is it different or some unique compared to other microbiomes? So I said three type of organisms, pinus tree in the bark, and there are two lichen species named Divinaria and Palmotrema. Look at this Divinaria is really highly attached to the substrate, but Palmotrema is loosely attached to substrate. I chose the bark sample for a epiphytic microbiome because it might be one of the origin source of the establishing lichen microbiome. So about the diversity, the alpha diversity was higher in this brown epiphytic microbiome than the lichen microbiome. And about the taxonomic composition, this bark harbored more abundant Basidiomycota compared to these two lichen microbiome. Then what about the beta diversity? We can see there are lots of shared ASB between the pine tree and lichen. And more interestingly, this pine epiphytic microbiome shared more ASB in the Dirinaria than Palmotrema. You remember that Dirinaria is highly attached and Palmotrema is loosely. And about the ordination, we can see these two lichen microbiome are like similar than each other, and they can be highly distinguished to the epiphytic microbiome. And with a dissimilarity analysis, it showed that this lichen microbiome has higher intra-host dissimilarity. It means that they do not share large ASB or fungal members even in the same species. And this similarity comparison between the microbiome said that this Pinus epiphytic microbiome is more similar to Dirinaria, like this one, than Palmotrema. And it is the most interesting result in this paper, I guess. When I see network structure of microbiomes, the lichen microbiome showed higher modularity and least nestedness. So what does it mean, higher modular and less nested? It means that the lichen microbiome harbors a very specialized cluster, and they do not share lots of fungal members. And lower diversity and higher modularity and least nestedness, it makes me think like there might be some effect of post-filtration in lichen microbiome. And when I found that each core microbiomes of these three fungal communities, I found this Pinus epiphytic microbiome shared 5 core ASB with Dirinaria, but not in a Palmotrema. So finally, when I saw the host specialist, it seems like compared to epiphytic microbiome, lichen microbiome harbors more plant-associated fungi. Yeah, almost less topic. So I guess many researchers investigating microbiome structure, they would have some common question that there is a microbiome structure and there will be host effect and the kind of locational or environmental effect. Which one is stronger? That is the study about that. So I collected 150 lichen in 3 distinct regions and extracted their climatic values from the World Climate Database, and it showed that these 3 regions have significant difference in climate. This is a step about the haplotype analysis. When I tried in a phylogenetic tree with using only one gene locus named ITS, internal transcript spacer, in the genus of that Palmotrema, it seems like that using only one gene locus is quite enough for a species identification. So I thought that maybe using this one marker for a haplotype analysis it can be good. So this 150 lichen, one species, are clustered into 16 haplotypes. And after discriminant analysis, these 16 haplotypes are grouped into 3 haplotype clusters. And as we can see, these 3 clusters have different phylogenetic distance and quite equally distribute to the 3 region. Then what about the microbiome structure? In a better analysis that more than haplotype cluster, then this location has more stronger impact on a lichen microbiome. Also in alpha diversity, there was no difference between the haplotypes, but difference in the location difference. In a taxonomic composition, there will be slight difference in the sky blue one. In the location B, there will be more abundance of Sodariomycetes. Then, okay, location or environmental factors affect this lichen microbiome structure. So I just wonder that which climatic factors can affect the fungal diversity. So after multiple linear regression, I found that the common fungal licheness is related to bio3 named isothermality and fungal diversity shown on there. It is related to three climatic factors. Then what about the fungal niche of this microbiome? So I tracked their troping mode with using fungal trait database and some lichen microbiome paper. So I found that there are three important groups that endolichenic, lichenic colors, they regarded as a core microbiome in lichen. And there are pathotrope, saprotrope, it regarded as a transient group in lichen. So I found that some negative relationship between this core group and transient group. So how can I make a conclusion with this climatic factor, diversity, and their fungal niche, different abundance? So with using structural equational modeling, I found this one. I really love this data, but it seems like they don't like me because what is meaning? But it is just the result of modeling, but I guess it is from because of the some different physiology of the group, like a transient group, pathotrope, and saprotrope. They are usually belonging to Sodariomycetes, and as you can see, they do not have much of melanin in their cell wall. But about this core group, they are really slow growing and highly melanized cell wall. That what melanin can do in this organism. Melanin can protect like from the UV or some heat or cold shock. So I guess when the isothermality, it means that when we can call it the higher isothermality, it is the higher fluctuation of temperature in the daily. So maybe, and there is a positive relationship of this core group and temperature, a newer range. So maybe big range of temperature, it is not good for this kind of group. And when it reduced in a high temperature fluctuation, it reduced this transient group and resulted in decreased diversity of lichen microbiome. Yeah, so too short about the four topics, right? And in summary, in a culture step, it was like a war against filamentous fungi. But when I used the small segments of lichen and using combination of some different nutrient-rich and poor medium, I can isolate slow-growing fungi. And about the comparison between the cultural result and amplicon sequencing result, that it showed that amplicon sequencing result does not cover all the range of culture. So maybe potential of cultural mix. And compared to the neighboring epiphytic microbiome, lichen microbiome showed less diverse, more modular, and less nestedness structure. And about the host or locational effect, more than haplotype, it seems like locational or some climatic factor affect the structure of lichen microbiome. Then what I did in my PhD is kind of fungal diversity based on culture-based, culture-dependent, and culture-independent. It is really similar to what I'm doing with Anthony in UH. The first project I'm doing is isolate super diverse yeast species. So yeast are really undiscovered and underrepresented taxa. And what about the unique and relatively undiscovered Hawaiian ecosystem? There must be a bunch of novel species candidates. So this is a short workflow that after liquid enrichment with doing high throughput culturing and with a nanopore with a high throughput sequencing, I'm gonna do isolate diverse yeast and identify. And there are lots of troubleshooting points, but I can show you one example. Filamentous fungi again. So it seems like they are really my enemy for my whole research lab, right? But yeah, I tried three approach. Like the first one is liquid culture. So in a liquid culture, many filamentous fungi show some gathering themselves and to make some solid structure like this one. And so I want to use this only liquid part. And second trial is acidified environment. As we know, yeasts are more tolerant to the lower pH, like a pH 4 compared to the filamentous fungi. But still, there are lots of filamentous fungi, and the last trial was just using a filter with size 10 microfilter. Most of yeast, their cell size like from 3 to 6 micrometer, and I guess it works. Look at that, how clear. And the second project I'm doing is animal microbiome. There are lots of study about the bacterial microbiome in an animal host, right? But really not many reports about the fungal community. So I want to compare in a big scale like a mammal, reptile, birds, and compare their microbiome and do some co-cladogenesis analysis like a copyrogeny. So our great collaborator in Honolulu Zoo gives me a bigger sample like a rhino, elephant, tiger to me, poo guy. So it was too short, but anyway, I want to express, I want to say thank you for support for my previous and current lab member. Thank you for listening.